I'm curious, do you think the biology of a T cell will be not identical, but will be comparable? You are going to find that elements are very similar. And often in this chapter, you will, or my notes will say things like, remember how the immunoglobulin worked. Well, this works a little bit differently, or it has these similarities. And so really, if you learn the pattern for one, you can, in effect, apply that pattern to the other and then note the differences. So a lot of like what you might do in an anatomy class is you learn the rule as a whole, and then you really focus on the exceptions. Here, you get to learn the pattern as a whole and highlight the things that are different between B and T cells. Now, we got all of this stuff. Just a very brief overview. I assume you all know this already. What does a T cell do? Or I guess, what part of the immune response does a T cell belong with? Yeah, it's an adaptive thing. What we will see is that there are some cells that are T cells that, yes, have elements of an adaptive response, but also act like an innate response. Remember, immunology is complicated, and the cardiologist said, take me, I don't want to hear this. So generally, T cells are an adaptive immune response. Their best function is to secrete lots of cytokines. That's most of what we'll talk about in chapter 11, or how T cells do their thing. There are some T cells that are really good at causing cells to die. Those are our cytotoxic T cells. Now, T cells see their specific antigen, and we know it has to be a specific antigen because these are an adaptive cell. It sees that through a neatly named receptor called the T cell receptor. More reminders. Where do you find the T cell receptor? On a T cell. And a few other cell types that have features of T cells that we'll see near the end. A reminder, the adaptive immune response has large amounts of diversity, 10 to the 9 or so possibilities for different antigens that a T cell could see among your entire T cells. That is in addition to the possibilities that a B cell receptor could see. So you see we have T cells and B cells to get, they can each see 10 to the 9 antigens, but many of those antigens we want our B cells and our T cells to both see. All right, so we've got lots of possibilities. And just like B cells, if I were to take one B cell from my blood, how many antigen specificities would that B cell have? How many different antigens could it buy? Just one, right? One B cell equals one possibility. For T cells, the same holds true. One T cell can see antigen, or can see one epitope, one type of antigen. But among all of our T cells, each one is going to see a different antigen, so we say that they are clonally distributed, right? Among all of your T cells, they're all going to see different ones until a T cell gets activated. And then it will undergo that clonal selection hypothesis, where that one T cell will replicate and it will keep generating more and more T cells that have the identical receptor specificity. Now, we really skimmed it. We dumped the immunogenetics chapters because, in my opinion, those are the chapters that are the most boring. And if you like genetics, we can do an independent study that you'll teach me how it works. But the events that occur when you build your T cell receptor are similar to the way you build your B cell receptor. So you have VDJ gene rearrangements, you have different segments. So instead of having 10 to the 9 genes for T cell receptors, you have pieces that can be mixed and matched. So there's similarity there. Now, the major form of T cell receptor is what is called alpha beta. So named because it's the most prominent, the most prevalent. When we talk about T cell activity, you are probably talking about alpha beta T cell receptors. Now, the alpha beta, each one of those is a different chain. And before I click ahead, if I were to tell you that TCR and immunoglobulins probably evolved out of the same gene ancestor, what superfamily would T cell receptors belong to? So remember, there's this super family that has these little domains, these little bumps. B cell receptors and antibodies belong to the Ig superfamily because they have these Ig fold domains. The T cell receptor also belong to that superfamily. And so when you see it sketched out, they resemble the structure you would see in an immunoglobulin or in a Ig superfamily member. And so alpha beta has two chains, an alpha chain and a beta chain. And on the membrane, they are linked together with disulfide bonds and they get expressed. And this is what they look like. Does that not resemble the bones of the structure we saw with B cell receptor? Got the lumps, these intra-chain sulfide bridges giving us these little globular domains, Ig folds. Now, just like our B cell receptor, at the end of those antibodies, you had an antigen binding site. T cell receptors have an antigen binding site as well. When we're comparing, or when we're talking about the alpha and beta chains, they're 40 or 60 kD. So they're much smaller than our Ig domains. But just like our immunoglobulin domains, they have, each chain has a variable region. So what do you think the variable region is going to do? Right, yeah, bind antigen. The part that can be the most variable makes sense to be the part that can bind any number of these antigens, right? So there's a similarity there. The variable region of our T cell receptor is analogous to the variable region of our immunoglobulins. They'll also have a constant region, which is more stable. Each chain only has one constant region though, and that's why these chains are much shorter. Just like our immunoglobulins, these variable regions have 3 CDRs. What do we remember happens in these complementary determining regions? You get these events, hypervariability. The entire variable region is subject to change, but there are 3 particular parts that are above and beyond their capacity to be different. What's cool is that the variable region on the beta chain, this piece, actually has an extra CDR. It has a 4th. And that 4th, that we skipped the chapter to talk about MHC, but it has the 4th because that 4th CDR region on the variable portion of the beta chain actually interacts with MHC. So it has a little bit extra diversity that's done. I'm sorry, it doesn't interact with MHC. It binds these things called super antigens. And we refer to those, I don't remember at which point. These are the things that can activate T cells in the absence of any stimulation. So toxic shock syndrome is caused by a toxin that Staphylococcus species produce, and they can activate T cells inappropriately. And so the question gets asked every time I teach stuff on super antigens, why? Probably super antigen, the ability of T cell receptors to be bypassed, to just have them activate, is probably a defense mechanism. That toxin is bad. We want a very robust response. But as our immune system has gotten more evolved, it has now become a detriment, right? It's just still present because I guess evolution works that way. So we have these super antigen binding sites which evoke pathological responses, all because the normal checks and balances get bypassed. So when we compare immunoglobulins to T cell receptors, we can compare them in 4 key places. Ability to recognize antigen, their valence, so kind of their overall structure, how they're put together, how they're stuck out on the membrane, whether they're secreted. I'm curious, are immunoglobulin, are those secreted? If you activate a B cell, what's the outcome? You're gonna make antibody, and antibodies have they are the secreted form of membrane-bound Ig. So Igs can be secreted. Do you think T cell receptors can be secreted? Yeah, I saw some shrugs, like, I don't know, maybe. No, they aren't secreted. What do T cells, when they get activated, secrete instead? They secrete cytokines. So we have a difference in the events that are related to an antigen response. T cells get activated to produce cytokines to cause other responses. B cells get activated to secrete things to do a direct effect. Right. So with antigen recognition, immunoglobulins are capable of binding a wide variety. This was chapter 4, I think, where we kind of compared the difference between the two. So they're able to bind carbohydrates, proteins, nucleic acids, lipids. An antibody can bind anything. And that's the nature of the way B cell receptors can see antigen. They don't need any help to see it. It just needs to be something that's facing out so they can actually have access to it. That means that Ig receptors can bind things that are linear. So amino acid 1, 2, 3, 4 can be the sequence. It can also bind things that are conformational, meaning the shape that they're in, how they are folded can be a way to recognize it. And Ig, because they can be membrane or secreted, they can see things in a variety of ways. Also, because they don't need any help, they can see things that are in the fluid phase. The alpha beta T cell receptors are restricted, meaning they can only recognize things in sequence. So a sequence of amino acids is what's going to be bound. They can only see that antigen if it's complexed with either MHC1 or 2. And what that means is that the antigen has to be processed. Think back to chapter 4. What's one of the characteristics of a good T cell antigen? Something that can be broken up, can be digested. There are differences in the types of antigens. We skipped the how are things processed. MHC1 antigens are smaller, 8 to 9 amino acids. MHC2 antigens can be a little bit bigger, 12 to 17, thereabouts. Now, notice for both of these, we're only talking about their size in amino acids. That reinforces the fact that T cells can only see protein antigens, because protein antigens are the ones that get degraded best, right? They can be taken in and chewed up and presented well. Now, the T cell receptor does both antigen recognition, but it also recognizes the MHC. Both of those are the parts that matter. And if you look at the CDR regions, this image helps illustrate that. 3 CDRs that form that binding pocket. CDR3 is principally the one that sees antigen. CDRs 1 and 2 predominantly see MHC. You need all 3 to recognize. So you need that really nice handshake. And not only is the handshake important to activate the T cell, that's also that fact that MHC is expressed is one way that you have cells that can destroy inappropriately infected cells. Right? If they're not expressing MHC, or they're expressing too much, or it's altered, we have cell types that can go and get rid of them. Now, another place to compare is in its valence and conformation. Immunoglobulins have 4 chains. What do we call those chains? They have 2 of each. Yeah, heavy and light. 2 little ones, and the light chains have a variable and a constant region. And then the heavy chains have 1 variable and then how many? 3 or 4 constant regions. And they're all linked together. And because it has 4 chains, it also is what we say bivalent. How many antigens can a B cell receptor or an antibody bind? You bind 2, right? They're like little Y's sticking out. Now, what's also cool is that B cell receptors and antibodies have that hinge region present. They have that flexible portion that gives them the ability to bind antigens and orient themselves. That also gives them function. The hinge region is how complement's activated. That's how some receptors are interacted with. Unlike immunoglobulin, T cell receptor only has 2 chains, an alpha and a beta at this point, which means it's monovalent, so it can only see 1 antigen in 1 place. And because it has no hinge region, we consider it much more rigid. And that sounds bad, but the rigidity of a T cell receptor actually causes it to be much stronger at interacting with other cells, because the T cell receptor needs to see MHC, and both of those molecules are rigid. So it makes a nice firm lock when they match up. A nice little quote that I pulled because I think it helps to illustrate. Remember, we talked about how antibodies get digested out. We can create these pieces. And there's one way to digest them where you generate these FAB regions. T cell receptors are akin to an FAB region of an antibody. You got an antigen binding site and you got two constant parts, and that's it. Now, simply put, there's a difference in secretion. Immunoglobulins can be secreted or membrane bound, depending on the state of B cell activation, and T cell receptors are not secreted. See, that was easy. Oh man, receptor changes. Does the IG during a B cell response, does it ever change? Does it change for the better or the worse? Yeah, recall we have those series of events, somatic hypermutation. It still binds the same epitope, but it starts to bind it more tightly. Well, that's the hope. You bind it more tightly. Affinity maturation is where you only keep the ones that bind better and you get rid of the ones that don't. And then class switching. The genes for your immunoglobulins will change. Instead of expressing the mu chain for your heavy regions, you could express alpha, gamma, epsilon. And that is unique to the function and the activity of B cells and their IG genes. T cell receptors, after they have seen antigen, they don't change. So they undergo no post-translational or they undergo no somatic changes. Their antigen receptor affinity is the same whether they are activated or not. And so what we say is after they come out of the thymus, the T cell receptor is fixed. It does not change at that point. So those are how they compare. So we're only going to see the T cell receptor on the surface. Just like with the B cell receptor, does it have other things associated with it? You have the membrane stuck, or you have the antibody stuck on the membrane. Is there anything else in the vicinity? What else do we have? We have IG alpha and beta. What do they do? I heard a hesitant signal, right? The immunoglobulin is who sees the antigen, but you need to take that message on the outside and affect the cell on the inside. And IG alpha and beta are the signaling molecules. And then you have the B cell co-receptors, which include CD21. Remember, I'm a complement guy. What does CD21 bind? Something related to complement, right? Evidence that complement has been activated. That's what CD21 binds. So there is an analogous group of structures related to the T cell receptor. On the cell surface, we've got our T cell receptor. That's who's binding the antigen. You will also have an associated, very tightly bound, but it's non-covalent. You'll have an associated structure called CD3. Now your text, and I've tried to keep true to this, but I'll tell you what the text says, and then I'll tell you how I treat it. CD3 is the signaling molecule. In your text, it gives CD3 as you have 4 different chains, plus it says you also have an associated zeta chains. For the purposes of my exam, CD3 is the signaling molecule, and that includes all 6 chains. So if you were to be asked a question, who does the signaling inside the cell during T cell receptor activation? You would say CD3. And we would just understand that you know that the zeta chains are a part of that. There's some distinction as to how you group those together. Just like with IG alpha and beta, when CD3, or I should say, when the T cell receptor sees the antigen, that will cause a change in CD3 so that they shift just a little bit differently. And that allows the activation of these regions called ITAMs. What does the A stand for in ITAMs? Activating. And so the ITAMs are these tyrosine residues. You can see you have a lot on the zeta chain, so they are good at getting this signaling going. These ITAM regions will get phosphorylated. That will allow other proteins to come, and that will transduce the signal signal, so the cell gets activated. So the analogy here is the T cell receptor is analogous to the immunoglobulin, the membrane-bound Ig. CD3, the signal transducer in the T cell, is analogous to Ig alpha and beta, the signal transducer in a B cell.
Which ones are activating?
So that would be where's my finger? So once we've seen antigen, CD3 gets activated. That will stimulate it could be, yeah, it's going to stimulate the program that will lead to cytokine production. And then depending on all the other events that we'll talk about in chapter 11, you could get Th1 or Th2, but you need CD3 to take the message from the outside and deliver it to the nucleus. Yeah, good question. What's also interesting about CD3, yes, it's a signaling molecule, but during development, it also plays a role in guiding the T cell receptor to the surface. So if you knock out CD3 in mice or human lines, what you will find is that T cells no longer express TCR on the membrane. So not only do you need CD3 to do the signaling, you also need it to put it in the appropriate place to see antigen. So what we say is that T cell receptor and CD3 are always together. You will never see them separated. So much so that a good marker for just general T cells is CD3. Now, beyond, so we have the T cell receptor, we have the antigen binding portion, which is the T cell receptor, and we have the signal transducer, which is the CD3. You are going to also have co-receptors, just like the co-receptor in B cells. They were there to make activation better, right? CD21 sees evidence of complement activation. It enhances the signal, so it takes less work to get the B cell activated. The co-receptors in T cells are CD4 and CD8, and they serve a similar purpose. What ends up happening is that your mature T cells will never express both. You will have alpha beta T cells that can only be CD4 positive, or alpha beta T cells that can only be CD8 positive. There was a study that came out, oh man, it must have been 7 years ago, and they were presenting evidence that they could, if you looked and did the right kind of cell analysis, they could find some T cells that did express both. And that was the only thing that ever came out about it. So to the point that, yes, that's an interesting tidbit, like maybe sometimes that happens, but it seems functionally it might not matter. I would argue that might just be an artifact. So for our purposes, can a T cell be in, a mature circulating T cell that we could pull out of your blood? Can it express both CD4 and CD8? It's got to be one or the other. And we will see the reason for that are the events that occur in the thymus. Now, if we were to go into your thymus, we could find T cells that do express both. So if I were to give you the hypothetical, I have found a T cell that expresses both CD4 and CD8. Your mind should start jumping to the fact it's not mature. It's not ready to do its thing. It hasn't been selected for. It's too young to know know what's good for it. Now, those co-receptors, CD4 or CD8, are both glycoproteins. And what's interesting about them, they do their enhancing in a way similar to the B cell co-receptor. They don't see antigen directly. CD21, does it bind antigen? No, it binds complement activation on the antigen. CD4 and CD8 bind to the MHC molecules. It's an additional, it's like shaking hands and then also clapping the person on the back. And it's like, oh, that really means that you like each other. Where a handshake, maybe not. They are glycoproteins, so they're sugar proteins. They have CD4, you will see drawn as having a single chain. Some texts do this. This text doesn't do a great job representing them so that it's obvious. So CD4 is just one little chain that interacts with MHC. CD8 actually has two chains. And because of the nature of the way they interact, they are restricted. And that's a term we'll come to when we talk about thymic events. CD4 T cells can only see antigen that's complexed with MHC class 2. And part of that is because the co-receptor, CD4, can only interact with MHC2. CD8 can only interact with MHC1. So that means CD8 positive T cells can only see antigen that's presented by MHC class 1. So we have a T cell restriction so that our cytotoxic T cells see antigen in one context and our T helper cells see it in a different context. Now, these CD4 and CD8s, you would say, oh, well, they're expressed on T cells. They must be good markers for T cells, and they are not bad. Like most of the things we will come across express those, but you can find CD4 and CD8 on other cell types. So if I said I identified a new CD4 positive cell, it has to be a T cell, right? And you could say, well, no, maybe not. Like there's a chance that it could be, but maybe not. So those co-receptors, because we have T cells that can only express one or the other, they give us the ability to separate function. T cells that express CD4 are related to cytokine secretion, T helper cells. Those that express CD8 are related to cellular killing, or cytotoxic T cells. What's cool is that if you are healthy, you don't have, we say healthy somewhat surreptitiously, if you don't have an active pathological viral infection, or bacterial, I guess, you will have twice as many CD4s as CD8s, which actually kind of works in your benefit because the CD8s are the ones that can cause cell damage much more quickly because they go in and they destroy it. But if you get an active viral infection, your CD8 positive T cells will go up because you have a viral response. We need to destroy the infected host cells. Let's get more of the cells that do that. There are cases where you can increase CD4, but I went with the one your textbook kept, which was, that balance can change. So in the case of HIV infection, the higher the viral load of HIV, the lower your CD4 T cells get. Viral load and CD4 counts move together so that if you have less than 200, we'll just use units of CD4 positive T cells, that's a sign that your HIV is progressing towards AIDS. Right, you can use that clinically. Now, why is it? We will touch on that in one of the later chapters. HIV virus uses CD4 as a way to get into those cells. And when it does, it can promote their destruction. So there are three important functions. Well, I guess two important functions and then one, the ability to use as a viral entry point. So we'll start with the one that's already obvious. Co-receptors, as we learned in B cell receptors, co-receptors enhance the signal. They make it easier to get that cell activated. CD4 and CD8, when they see MHC 1 or 2, whichever they're supposed to see, they themselves can activate kinases that can drive signal transduction. So the T cell receptor alone could cause 10x signal transduction. If the co-receptor also does it, it could cause 100x. It makes it much more likely, analogous to what you see with CD21 and the B cell receptor. Now, those co-receptors also play a role in just holding the cells together. To get all of these activation events, you need engagement. And so by having CD4 interacting with MHC class 2, which is only expressed on antigen presenting cells, it holds it together longer, making activation more likely. CD8 interacts with MHC class 1, which is also expressed by antigen presenting cells, but also any nucleated cell. And then the co-receptors can act as viral entry points, HIV being the best example. All right, so they enhance signal transduction and they hold them together. Now, we also need co-stimulation. So we need to see antigen, we can enhance that with our co-receptors, and then we need to make sure to get full, complete T cell activation that all of the rest of the message is strong. And so we're going to take naive T cells, which means they are ready to be activated, but they've never seen antigen before. They know what to do, they've just never done it. The first signal that this naive T cell needs is to see the peptide and the MHC. So we need T cell receptor engagement. The co-receptor will interact. The second set is something that occurs in a different place. Is co-stimulation. This is not linked to T cell receptor binding. And that includes, here we go, certain co-stimulatory molecules. And I have these two on a slide together because they work in opposites. They work, they are contradictory. That's what I'm going for. So T cells can express a co-stim molecule called CD28. CD28 interacts with a group of proteins called the B7 family. Depending on the picture I put, you might also see that called CD80/86. There's just a collection of proteins. The B7 family are going to be molecules, they're going to be ligands for the CD28 that are expressed on APCs. For a T cell to get its full and complete activation, CD28 needs to be engaged, because what that does is it induces the T cell to produce IL-2. And if you recall from chapter 12, IL-2 is a cytokine that drives T cell proliferation. The opposite to that is CTLA4. It is related to CD28. They come out of the same family. It also interacts with the same B7 family. What happens here is it's a negative signal. So the longer a T cell is active, and it's interacting with its T cell receptor, it'll start to produce more and more CTLA4, to the point that CTLA4 binds instead of CD28. And what do you think that does to the T cell? It turns it off. Why would we want to turn off a T cell? Yeah, we don't want inflammation to occur indefinitely. T cells, by the virtue of the things they express, they can be overcome. They can have a time limit. In theory, yeah, there is a magnitude that you could reach. Now, in practice, what can contradict the function of CTLA4 is everything else that's happening, right? So if you also have neutrophils and B cells doing it, they can turn down CTLA4 expression so that you don't get that inhibition. And what's interesting is that cancer cells use CTLA4 as a way to avoid the activating T cells. They will start, actually, did I bring it? Yeah, I did include it. Cancerous cells can actually express CTLA4 on purpose to try to turn off T cells so they don't get activated against them. So I borrowed this from the other text, and that's where in 2018 there was an anti-cancer drug called ipilimumab. It is a CD28 antagonist, CD28 agonist. You can treat people who have certain types of cancers with this antibody, and it binds to CD28, stimulating T cell activation. Because many cancers avoid that by blocking CD28 signaling. And a guy from Mizzou, I think it was him, yeah. He did some of the chemistry side, and I think he shared part of that Nobel Prize of chemistry. Yeah, that sounds right. So with these co-stim, if you have a T cell, it has seen its antigen, and there are events that occur. So the co-stim events will only work if it's seeing antigen that's presented from the right place. And if that happens, you get an activated T cell to produce cytokines that'll cause death, inflammation, destruction, all that. If that T cell now is starting to express CTLA4, that's what interacts instead of CD28, and that T cell becomes anergic because it's being turned off. And anergy is just making a T cell or making a cell do nothing. And there are some restriction events. If a T cell tries to see an antigen that's not being expressed by the right group, they don't express the co-stim molecules, T cell won't activate. So there you have some proofing in the background. There is another co-stim molecule to talk about, and it's called CD40 ligand or CD40L. It interacts with CD40 that's expressed in other locations. And what this serves to do is enhance the signal. So CD28 binding is good. If you get CD28 and CD40 ligand binding, it's even better. So in addition to co-stim, we're going to express some things that also are necessary for adhesion and homing. Because what do lymphocytes do? Unless they are activated, they do what all the time? Are they constantly moving? All right. Why are they constantly moving? Yeah, because you have a lot of place these antigens could be. And so parts of the immune system concentrate antigens in particular locations, and then you have lymphocytes that can circulate around. Every 12 hours they can migrate through the entirety of the body. But when they cease, they need to know where to go, but also if they interact with antigen, they need to be able to stop. And so this is the balance of adhesion and homing. What's interesting about these molecules, none of them are specific for T cells. It's a lot of what we talked about with B cells. They kind of provide addresses. So certain tissues express a collection of receptors and molecules that will cause the attraction of certain cell types. Now, this types of adhesion, one of the things it does is it allows cells to enter the appropriate location so they can see antigen. Adhesion also slows them down, which increases the amount of time for them to see, is this the right antigen? And this is especially true on dendritic cells. Now, we have 3 key molecules to talk. We have one called CD2, and I borrowed this from another text. We avoid this terminology because it's complicated, or it can be complicating. Immune cells form things called synapses, just like neurons. And really the purpose is very similar. A synapse in an immune cell is to allow consistent interaction so that you get activation. What CD2 does is it holds the T cell together with the cell that's trying to activate it. And it does that by interacting with LFA3. You have another molecule expressed by T cells called LFA1. It binds to a ton of things, ICAM1 being one of these groups, and that helps it hold on to it. T cells can also express ICAM3, which binds to LFA1 expressed on the cell trying to activate it. So the moral of the story is something like CD2, if you were to knock that out, I give you a patient who does not express any CD2. Purely for T cells, what could be the complication? Yeah, you'll still have some, right? Like odds are you'll still have some, but you're going to see a reduction in the amount of T cell activation because they're not going to be able to hold together as long, and time is what matters. You need them to hold together so that the signals can activate. Now, we talked briefly about homing. We will keep it fairly brief here. T cells need to be guided to particular locations. It's very similar to what you see with B cells. It is that it creates these addresses so that the gut mucosa has a particular type of address versus a lymph node. And what this does is it allows T cells the opportunity to circulate. And if they don't see antigen, they'll continue to circulate. These naive T cells will move through. They have a collection of things that they use to enter into lymph nodes. One is L-selectin, and the L here stands for lymphocyte selectin. It binds to things expressed by the HEVs, which is the way that our lymphocytes leave the blood and enter the lymph node. They also migrate in response to they express CCR5, which is a chemokine receptor, so that these T cells can migrate based on chemokine concentrations. If it gets activated, it will change what types of homing molecules it's expressing so that it stays put. It gets activated, and then it leaves to go back to the tissue that activated it. So if my body is responding to a skin antigen, my T cells will eventually leave the lymph node as activated cells and go back to that skin place. Why would they go back there? Because that's where the problem was, right? They get activated in the lymph node, they migrate out so they can go back now as super active, strong pummelers. And I have the example of gut. So we spent all of our time at this point talking about alpha beta T cells. There is a really, and I don't know if this is the paper I picked for you. I had a collection that I was going to choose from, and I don't remember where I fell. So there's a collection of T cells that are called gamma delta. Gamma delta T cells are a minor population because they have T cell receptors that express a gamma chain and a delta chain. Whereas most T cell receptors are what? Alpha and beta. And what we call these T cells that express gamma delta T cell receptor, we call them gamma delta T cells or GD T cells or gamma delta T cells. Now, gamma delta T cells still have those T cell receptors associated with CD3. So is it still true that CD3 is a good marker, a good way to say we have a T cell? Yeah. So no matter which T cell receptor type we have, CD3 is still present, still a signal transducer. The exception with gamma delta T cells is they typically don't express any co-receptor. No CD4, no CD8. There are exceptions that I included. I don't care that you know that exception. Now, where you find gamma delta T cells, they tend to be well associated with the barrier. You see these a lot in tissues related to epithelial membranes and the mucosa. You find very little of them in the blood. And actually, to emphasize that, of all the lymphocytes, or I guess it shows you where they are, but also shows you how few of these there are. Of all the lymphocytes you would find in an epithelial surface, only 10% in normal conditions are going to be these gamma delta T cells. As you start to migrate into the intestine, you start to see that percentage, that proportion climb up. Gamma delta T cells, again, they have these T cell receptors that have a gamma chain and the delta chain. They both express the same V regions and C regions. So they have a variable and they have a constant. The antigen binding regions are still the variable portions. So structurally, they're fairly similar in the way it's laid out. But what's interesting is these gamma delta T cell receptor genes have fewer segments. So there is less antigenic possibility in your gamma delta T cells. What does that start to remind you of, maybe? What types of receptors have we talked about that have less diversity? Innate receptors. If you are binding fewer things, odds are you're probably binding things that are a little more common. Right? And that holds true. Gamma delta T cells have elements that are both adaptive because they have a T cell receptor, which is itself binding something specific, but then they share those elements that are also related to an innate response, meaning gamma delta T cells, they're at the barrier, so they activate quickly. They are good at producing inflammatory cytokines, things like interferon gamma in response to virus or TNF in response to anything. They can be cytotoxic, and interestingly, gamma delta T cells do not remember. They seem to respond to the same antigen, the same magnitude each time. Do those four traits sound similar to what we would expect an innate cell to do? Except they're doing it with a specific antigen receptor, although it does not bind the same diversity as our alpha beta. So what is it then? What is it binding? Turns out it's binding things that are small non-protein molecules, especially things like phospholipids. I'll give you three choices. Alpha beta T cell receptors are good at binding proteins, carbohydrates, or lipids. Proteins. Gamma delta T cells are not like that. They are good at binding, especially phospholipids or other small non-protein things. And you call these a collection of antigens called phosphoantigens. There are some similarities between them. And what these typically boil down to are proteins related to cellular stress. If you are expressing phospholipids that a gamma delta T cell can see, odds are something is wrong with you. Right? You have experienced shock or stress, or you are under attack by a pathogen. The gamma delta T cell will see that and will evoke a quick immune response. Now, in thymic development, gamma delta T cells arrive first. If we were to start to map these out, you have 3 pathways you can take. Really, the line should be kind of here. And they emerge in the thymus before alpha beta T cells have matured. And because they come first, that's a suggestion that they are probably more primitive. And they also probably play a role in helping to protect neonates. You start to see these, you don't have many T cells as a neonate, right? Your thymus isn't fully developed yet. But you can start to find gamma delta T cells in the mucosal of the intestine. Redundancy. The more things you can have, the better a chance you have at minimizing complications, like reducing invasion. Yeah. And that's true too, an evolutionary standpoint. If you start to trace these back, lesser evolved mammals they'll have different ones, but their alpha beta T cells are much less developed, and their gamma delta T cells seem to be the one that matters. So it's almost like you're finding remnants of the march towards adaptive immunity, which you're going to have some of that redundancy early. Okay, we have alpha beta, most common, gamma delta, smaller proportion. They act a little bit weird. So what about these genes? We're not talking about TCR genetics, so this is the abridged version. There is an organization to it, and what we do is allow it to match up to what you have already talked about and at some point will remember, is the organization for the genes related to immunoglobulin. So the alpha and gamma chains, alpha and gamma, are analogous to your light chains. So their genetics, they're going to have V and J regions. Smaller chain, fewer regions to pair together. Your beta and delta chains have 3 segments, the VDJ. And if you think back to B cell receptor diversity, right? We had those rearranging events. You take these different pieces and you stitch them together, and that gives you your diversity. These genes are also organized so that the chains are restricted, meaning you can only express alpha with beta, and you can only express gamma with delta. So you can't have a T cell receptor that is alpha delta. The way the genes are organized, it is prevented from occurring. I call this a restriction. It's the idea that these constant regions, some of them allow interaction with the co-receptor, but they don't have an effector function, right? Our immunoglobulins, when they're secreted, that FC portion can activate a macrophage or stimulate opsonization. These aren't being secreted, so those constant regions aren't inducing an effect. And then our alpha and beta genes have significantly more diversity. There are 40 to 50 gene segments possible. And then if you start to go through the math, that it's not exponential. There's some word for it. You use the sigma value. But you get lots more diversity than the number of segments you have for the variable regions for the delta and gamma, which only have 5 to 10 different pieces. So you can only rearrange those in so many ways. And I've added this, but I will not ask you anything off of this. But for me, it helps to see this, to see how they're organized. And what you see is alpha genes and delta genes are actually bundled together. So they are on the same locus. So that when you express alpha, that prevents delta from being expressed, which helps to restrict it so that alpha and beta can only go together and gamma and delta can only go together. Now, the types of diversity you get and the events that lead to that diversity are very similar. In fact, the same enzymes that are used to rearrange immunoglobulin genes rearrange T cell receptor genes. And I don't have those illustrated here, but they're a collection of things called RAG1 and RAG2. And what they do is they mix around the genes, they are who drive it. So if you were to knock out these recombinase enzymes in an individual. Not only will they not be able to produce diverse B cell receptors, they also won't be able to produce diverse T cell receptors. And you have a summary of T cell diversity. You have multiple VDJ, you have multiple pieces that can be rearranged. You can put the chains together in different ways, like a slightly different alpha can go with a slightly different beta, but can an alpha ever go with a delta? No, we are restricted so that alpha, beta go together, gamma, delta go together. There is what is called junctional and insertional variability. The moral of the story here is CDR3, that hypervariable region, can be influenced in the TCR to bind the antigen slightly differently. But the event is once that the final point is once the T cell receptor is established, once the T cell has passed all the checkpoints, that T cell receptor never changes. Can the B cell receptor change after it has passed all of its checkpoints? Right? Yeah, it can somatic hypermutate. It can, yeah, it'll do affinity maturation, and it will isotype switch. So during development, where does this occur for T cells? The T stands for thymus. So T cells, because they're a white blood cell, arise ultimately from a hematopoietic stem cell. In an adult, where do we find all our hematopoietic stem cells? Bone marrow. That's where they are. That's where they house. That's where they turn over. So after we're born, through post-birth development, bone marrow. Recall that when we are a fetus, especially early, they derive from our fetal liver, and some before that from a yolk sac, which is cool. Now, what will become the T cell lineage comes out of something called a common lymphoid progenitor, or a CLP cell. So as a cell differentiates, it can keep going down, but it can never go back up. So if we generated a common lymphoid progenitor, could it give rise to a hematopoietic stem cell? No, because we're getting more and more specific. You can actually find some of these CLPs that circulate through the blood, because what you are catching them doing is leaving the bone marrow and moving to the thymus. And because we're catching them to the thymus, what we know is occurring in the thymus is the development of T cells, and this makes it a primary lymphoid organ. Here, T cells are going to adapt the characteristics that make them T cells. They're going to get their diversity of T cell receptor, and while in the thymus, you're going to see significant amounts of developing T cells proliferating. Now, your thymus is also a site of lots of proliferation, but also lots of death. Upwards of 95% of all of these developing thymocytes will ultimately die. And why do you think they die? I'm glad you don't know, because then that makes the rest of this interesting. They die because either they don't bind the things they're supposed to, or they bind too well things they should not be binding to. Right. Now, as you get older, your thymus gets smaller. When you dissect a cadaver, most of those are advanced in age. You're not going to see any thymus. What you are seeing instead, if anything, is fat. Why does it do this process called thymic involution, which is the opposite of, which is related to devolution, but also not good? Why does it go away? Speculated and maybe proved, I didn't look into it, that as you get older, you start to produce more and more steroid hormones, right? That's what drives secondary sex changes at puberty. Do you know what you give to somebody who is having an inflammatory event? Like I do a lot of watching of sports. What do you give an athlete who's got knee pain or knee inflammation? What do you stick in there? You give them a steroid shot, right? Cortisol or corticosterone injection. That inhibits an immune response, or at least slows it down. So as you start to, so steroids block an immune response, probably contributing to the fact that your thymus gets smaller, because now you have lots of testosterone and estrogen circulating. And that's okay, right? What they find is that you can do this in mice. You can take the thymus out in a mouse that's very young, and it will show a diminished capacity to make T cells. It'll still make T cells, but they won't be as diverse because you took out the place where they're being developed. What do you think happens if you take the thymus out of an adult, one that's farther along in its age? You think it'll have the same consequence? You'll still see a change, but it's much less significant because most of those thymocytes that have been developing have already developed, and T cells tend to be long living. So once they're made, they just kind of circulate and bump around, and the bone marrow can accommodate for some of that development too. So when we say the word thymocyte, a thymocyte is not a T cell, but it is on its way to becoming a T cell. Thymocytes are little babies. They're learning the ropes. They're undergoing education. And what these thymocytes are going to do within the thymus is interact with a network of different cells. One of the most important are things called thymic non-lymphoid cells. Principally, these are epithelial cells. What these epithelial cells do is they express both MHC1 and MHC2. So they are acting like antigen presenting cells. You will also have some dendritic cells that are present. They themselves are also showing antigen in a way similar to any other dendritic cell we've already talked about. And the function of these thymic non-lymphoid cells, what they're up there doing is one, filtering out, doing the selection, who's good, who's bad. They're helping to drive those signals, but they're also making lots of IL-7, which is making these thymocytes live for as long as they are appropriate to stay living. I put it at the beginning because it's a great flow chart showing you the events that are going to occur. We have this common lymphoid precursor. It's going to leave the bone marrow and go to the thymus. And then at this point, now that they're all that orangey beige color, this is all occurring in the thymus. So what are the events that are going to occur? First thing is we're going to have a double negative T cell. What do you think it's double negative of? Co-receptors. Double negative T cell expresses neither CD4 nor CD8, hence double negative. Now, these double negative cells, once they hit the thymus, they're going to start to proliferate, right? We're building our numbers up. They don't express anything yet. They also do not express T cell receptor genes yet, but once they enter into the thymus, they receive the stimulus so that their T cell receptor genes start to become active. This is why we can produce gamma delta T cells first, because in this double negative state, here is where our pathway, are we going to be an alpha beta T cell, or are we going to be a gamma delta? If the gamma chain is produced, that cell is committed to becoming a gamma delta T cell. And what we're going to focus on is mostly the alpha beta, because that's the one that's most prevalent. So we have these double negative T cells. They're starting their TCR stuff. The next stage of development is what is called a pre-T cell. In a pre-T cell, what has happened is that the beta chain of the T cell receptor, that beta chain has been rearranged enough that something fruitful is being produced. In B cells, they have a pre-B cell receptor. Do you remember what part of the immunoglobulin is expressed in? And I will tell you, it's the first antibody that ultimately ends up getting produced also. The first antibody is which? IgM. The pre-B cell receptor, the first piece expressed is the mu chain. So why express one part and not just express the full thing altogether? Right? In the B cell, if that mu chain is not right, you have one more chance to fix it, but you haven't invested in doing anything else. Same is true in the beta chain. If you express a beta chain that sucks, you can try the other chromosome or the other allele on the other chromosome pair. And if it sucks, what do you think happens to the T cell? Yeah, it'll undergo apoptosis. So by expressing this pre-T cell receptor, you get you have a checkpoint. Are we on the right path? Like, is this going to be valuable before we waste all of our time? Much like the pre-B cell receptor, when you express the beta chain in these pre-T cells, it gets paired together with a placeholder molecule called a pre-T alpha and CD3. So in B cells, their pre-B cell receptor expresses the signaling molecules. And it's thought that is so that the cell knows, did we express a mu chain that works? Similar why we're going to go ahead and express CD3 now. Because it's taking the receptor to the surface, and then it can tell the T cell, did we do it right? And those that don't express a fruitful copy or don't express a beta chain at all at this point undergo apoptosis. So this is our first checkpoint, and we again have allelic exclusion. When one beta chain is expressed, you don't bother messing with the other one. If the first one fails, you'll try the other allele. Now, after the pre-T cell receptor is expressed, and it's expressed fruitfully, we undergo the process of alpha chain rearrangement. So we make sure one chain is good before we bother doing the other side. And this is why beta, you can only get alpha beta or gamma delta. Because once the alpha chain is expressed, alpha and gamma are found on the same locus. So if you express alpha, that's because you clipped out all of the other gamma chains. So you can only get one or the other. What's interesting, just like light chain, the alpha chain does not undergo allelic exclusion. So you make a beta chain and then you can pair different alphas with it based on how they're rearranged. And so that aids in the diversity. Once that pre-T cell receptor has had a successful signal via CD3, now we're going to start to express our co-receptors. And here we have a double positive cell. Double positive because it expresses both CD4 and CD8. And this represents, of the thymocytes in the thymus at this point, 80% of them are double positive. I'm sorry, not at this point. Of all of the living cells in the thymus, 80% of them are found as double positive. And so they're immature. You can call these alpha beta positive because they have the alpha and beta T cell receptor chains being expressed. CD3 positive because CD3 is always with the T cell receptor. CD4 positive and CD8 positive. But now we have this cell that basically can be any manner of an alpha beta T cell. So now we need to select. We've made a whole bunch of them. It's time to get rid of the ones that aren't any good, or at least get rid of the ones that have made it this far that aren't any good. The first event, or I guess the collection of events that will occur next, are all together called thymic selection. This ensures that specific features are kept. So the cells that are produced have these features. It also ensures that we get rid of cells that have features we want to avoid, namely strong self-reactivity. We also we can split these up into two sections: positive selection first, and then negative selection second. So in positive selection, our double positive cells are going to interact with those thymic epithelial cells. Recall those thymic epithelial cells express MHC1 and 2, and it has antigen in it. So what do you think the positive selection here is for? What event are we selecting? What event are we trying to keep? What we're after here is we want these double positive T cells, we want them to be able to interact with MHC and antigen. If it's expressing a T cell receptor that can't see antigen or the MHC, do we want to keep it? Because if you put that thing out in the periphery, it's not going to be able to get activated. And what this does is it results in the survival of that double positive cell. And interestingly, depending on what it interacts with, and it looks like it might just be, well, there are there are pathways, but I like to think it's just chance. A double positive cell has a chance to interact with MHC1 first or MHC2. And whichever one it interacts with first, that's the cell that it's going to become. So that after the positive selection event, we have committed T cells, meaning they will only express one receptor or one co-receptor. So to survive positive selection, what this actually means is you have to have some self-reactivity, because you need to see that MHC and you need to interact with it. This is what we would say now, a cell that passes positive selection is going to be an alpha beta cell that is educated, meaning it has some affinity for MHC1 or 2, depending on if it's a CD8 positive T cell, it will have seen antigen on MHC class 1. If it's a CD4 positive T cell, it'll see antigen on MHC class 2. If you want to hear a really poor way, I still, as a 15-year, however long I've finished my PhD, no, 8 years. I've had my PhD 8 years. The way I still remember which interacts, what is half of 4? 2. I still look at that and say, well, half of 4 is 2. CD4 has to interact with MHC class 2. It is stupid and pointless, and it's not logical, but it works for me. Because then if you do what is half of 8, that's 4. There is no MHC class 4. It can't possibly work. Yeah, you look into it too much and it falls apart. So what we say is that we now have self-MHC restriction. It's educated and it's restricted. The CD4 positive T cell can only interact with MHC class 2. Also happens that's the only thing it can express. And same is true with CD8. Now, of all of those, during this positive selection, of all of those double positive cells that reach this point, about 90% of them will have poor or no affinity for MHC and those antigens, those peptides. Here's the trick. To be a developing T cell, to actually make it, you have to bind a little bit, but we'll see in the negative selection steps, you can't bind too strongly. Because what will a T cell do when it's out in the environment? It's going to interact with self MHCs. So it's got to be able to do that some. And here are events for that. And if it does not interact, or it's very weak interaction at this point, it undergoes programmed cell death. The other set of events is we also need to get rid of those developing thymocytes that are going to bind too strongly to self-antigens or to self-MHC, because if those escape, they will start to see all of this tissue that it is programmed to know, or it is programmed to think we've got to attack it. So this is the steps of negative selection. Positive selection keeps those that can interact. Negative selection gets rid of those that are too strong. And what we say is that we are getting rid of high reactivity against MHC proteins. This can be expressed, or this is achieved by interaction with dendritic cells or those same thymic epithelial cells. And what this ultimately results in is central tolerance. We will talk in an upcoming chapter about peripheral tolerance. But anytime you see tolerance, think the ability to not attack self. Central means it's occurring in primary lymphoid organs, the thymus, and then bone marrow for B cells. And by passing this negative selection checkpoint, we have confirmed that we have low self-reactivity. So our CD4 positive T cells are not going to bind every self protein that they see. And our CD8s are not going to bind every self protein they see. So we see if they have too high an interaction, they die. But as a single positive cell, whether that's CD4 or CD8, they still, remember, they have to have some affinity. They had to bind those to pass out a positive selection. So we want those that have modest amounts. They then need to leave. And at this point, once you've passed negative selection, you have an alpha beta T cell receptor, or you have an alpha beta T cell that is either CD4 positive or CD8. Studies from mice and then later fairly well confirmed in humans. If you were to track, we can image a mouse and watch in its thymus, and you can track one cell and see how long it takes to do its thing. It takes about 3 weeks from when a when a lymphoid progenitor enters the thymus. It takes 3 weeks for it to get out as a mature single positive cell. Once it is past positive selection, it seems to bump around in the thymus for a couple more days, and it likely is receiving more pro-survival signals so that when it exits into the blood, it's strong enough to survive and circulate. And naive T cells, should they not see any antigen, they can live for years. Remember, our thymus eventually goes away, yet we still can mount T cell responses. Those T cells can be long-lived. As long as they are receiving nourishment, principally in the form of IL-7 and other cytokines from cells of the lymph node and the bone marrow and tissue, wherever they're migrating to. So we have these cells that have left. Let's summarize or revisit what are our characteristics. Of an alpha beta T cell, they can either be CD4 or CD8 positive. Because we are mature, we are ready to see antigen, can only have one or the other. The events that they will do are self-restricted, meaning CD4 positive T cells can only see antigen that is with, half of 4 is 2, MHC class 2. CD8 positive T cells can only recognize antigen that is expressed with MHC class 1. Our single positive T cells are also self-tolerant, meaning should they see self-antigen, they should have a very low probability of responding, because negative selection should have gotten rid of most of those T cells that are strongly self-reactive. Do you think that actually happens all the time? All right, we know autoimmune diseases are a thing, and often that's because you have self-reactive T cells or lymphocytes of any type are able to escape these selection checkpoints. And so the question can be asked then, all right, well, they selected using self-antigen, and then we kept them. Like, we have these, why is there, like, where did those peptides come from? How are they going to bind non-self antigen? And that's an interesting question that itself leads into an entire semester's long course. And that's studying the fact that MHCs in the thymus seem to have waffly antigen binding. Those MHCs in the thymus that are expressing a self protein out in the periphery probably express a non-self protein. So that it is redundant. I don't know. That's not the right word. Anyway, the MHC in the periphery, those same self proteins likely aren't present. And that is as confusing as it might seem. So they need to see self, but when they're out in the periphery, they're not going to see that same self stuff. It won't be expressed. Now, we will talk in the next chapter. Once they get out into the periphery, our T cells are going to further differentiate. Our CD4 cells are going to activate, and they're going to become T helper 1 or 2 or 17 or 9 or whatever. And those are examples of effector cells. Those are going to be doing some sort of function, some sort of work. We also are going to produce memory cells. And those are the ones that can be reactivated quickly. It's like saving a document after every significant change. You create little copies, and if at any point you need to start from a previous copy, you have it ready. Now, what about some other cells in the thymus? Because yes, most of them are T cells, but there are other things that develop in the thymus. Majority are alpha beta T cell receptor T cells, which can either be CD4 or CD8. We also will make a small population of gamma delta T cells. The other ones that might pop up, natural killer T cells or NKTs. These represent about 1% of all non-granulocytes in the blood. So if we were to lump together lymphocytes and monocytes, we only have 1% of all of those cells being these guys. These are rare. They express a T cell receptor, making them an adaptive cell, but they also express this receptor in mice excuse me, called NK1.1, which is a natural killer cell marker. Natural killer cells, are they innate or adaptive? They are innate, right? They see threats and they assume everything needs to die until they get a handshake that says, don't kill me, I'm fine. NKT cells are able to respond to specifically glycolipids. And NKT cells are unusual in that they see these glycolipids on a molecule other than MHC. And it's a molecule called CD1D. They also don't seem to recognize a whole bunch of stuff. They're kind of restricted in the amount of glycolipids they can see. And so they have functions of both adaptive and innate immunity. A type that we will come across when we discuss autoimmunity, next unit, I think, are Treg cells, regulatory T cells. Their name implies they regulate the activity of something. They actually regulate the activity of other T cells. I guess immunity in general, but T cells especially. They're a small subset, but actually, if you think about their function, 10% of all of your CD4 positive T cells being a regulatory cell seems kind of high. And what they do is they express CD25 on their surface. They have a specific transcriptional factor called FOXP3. And they secrete cytokines that turn off activity of other cells, and they express things on their membrane. Should they bump into an activated T cell, it'll turn them off. And there are two places that we see regulatory T cells being most active. That is against self-antigens. So they're going to turn off T cells that are responding against self, and they can turn down responses by cells that are responding against foreign antigens. And this gives us two populations of T cells. We have a collection called natural Tregs. These develop in the thymus. These are actually autoreactive T cells that do escape negative selection. But their autoreactivity is important because they are autoreactive against cells that are responding to self-antigens. The natural Tregs can see, oh, you are responding against DNA. I'm going to turn you off. I'm going to cause you to die, or whatever. The other type are those that you produce in the periphery, and these are called induced Tregs. And these principally are impairing T cell responses against non-self antigens. These are what you would generate during the course of an immune response. And that's some of what we will refer back to, this idea of peripheral tolerance in some chapter. And then the last cell, they're natural killer cell. And what's interesting about these, most of these, we talked about them developing in the bone marrow. That's where most of them develop. Although you get a really small population that pop out of the thymus, they come off before the TCR genes start to get expressed, and they are involved in early innate responses. Natural killer cells coming out of the thymus seem to, for all intents and purposes, behave the same way as natural killer cells that are made in the bone marrow. If they get an inhibitory signal, they won't kill that cell. Usually that inhibitory signal is like MHC class 1. If they don't get it, the cell will say, well, this is a sign that something's wrong with you. I'm going to proceed with my killing program. So natural killer cells really help control viral infection and malignancy. Natural killer cells, because they don't have a T in their name anywhere, do not express T cell receptor, but instead have a couple of molecules, CD16 and CD56. If we were talking about mice, we would say they also express NK1.1 from the slide before. All right, and with all that, now you are experts in T cell development, more or less. All right. Anybody have any questions over this? Probably not yet, I guess. So hopefully what you can appreciate. Oh, Brooke asked, so what exactly is the difference between NK and NKT cells? Well, one, what they do. NK cells are reacting against altered host cells. NKT cells are responding to to lipid antigens that could be from foreign things. They could also be from self. So they behave differently. Another very simple one is that NKT cells express T cell receptor. NK cells don't. NKT cells were discovered, it was in the 70s that they started to find these weird, they were like, oh, these NK cells are weird. They express T cell receptor. Yeah. And if you scratch it deep enough, you will discover a new thing. Cool. And some of it's trivial. And I'm done. If you want to log off, you can. But I'm going to complain for a minute because you all can't leave yet, or I guess you can. But it's going to take you longer to get out. And exams, yeah, will be released. Or my goal is to have them done by Thursday before we meet. And for now, we'll meet online on Thursday. But hopefully, I can get it face to face more. Awesome.
Recording stopped.
You remember that first manuscript? I think it was the first one you did that was like M1 macrophages versus M2, right? So especially myeloid cells, there are neutrophils that they'll say, well, these are N1 neutrophils versus these that are N2. And M1 versus M2, it's what you're producing. So M1s tend to be more inflammatory because they produce cytokines that are inflammatory. M2s seem to be anti or reparative. A lot of that is trivial, right? So is there a difference between those two cells? People in immunology who study that, if you look the literature, there are now M3 cells. They express slightly different cytokines. There are M4, there's MOX that recognize oxidized lipids. And you would say, isn't that just a macrophage that is seeing oxidized lipids? So if you want to make a name for yourself, become an immunologist, identify a cell that produces one thing different, and then claim it's distinct and new. And then you can name it whatever you want.